Hey guys. Okay, so we're gonna go over how to solve genetics problems. I'm gonna go over a couple problems from problem set 1, and then I'm gonna go over a couple from problem set 2, and then go work on those. And then I'm gonna put a key up for those on Thursday. Nope, sorry. I'm gonna put it up on, let's do Friday. I'm gonna put it up on Saturday. So those are due Friday at midnight. I'll put the keys up on Saturday, just so you can check your work and see if you're doing it correctly. Remember, all practice work is just for completeness. So you just have to attempt every problem and you're going to be good to go. And then Saturday, I'll post the keys and then you can actually see if you're doing it correctly. And if not, then we should schedule a meeting to figure out what's going on. Okay. So just flip to your next clean page. Oh, we never did a title page. Okay, we're in our next unit. It's called Unit 6. It's our last one. Inheritance. So essentially my plan is that this week we're going to do genetics, and then next week we're going to do gene expression, and then I'm going to be done teaching new content. And so AP testing starts the week after next. And so I wanted to be done teaching content by the time your AP tests start, even though ours isn't until the 18th. So I'm going to finish teaching content next week. And then the following week, the week after that, I'm going to be posting review videos from the content from quarter 3. So that was before the midterm. So I'm going to just do run through reviews, and I'm going to post those on Classroom. Sorry, not Classroom. I'm going to post them on our Google Doc. So you guys can just access those when you have time throughout the week, essentially. And then our AP test is on the 18th. And then I'm probably going to give you a bit of a break after that. And then I'm going to figure out your quote unquote final. And then I'll just give you like the rest of the school year to pretty much work on your final stuff. So keep in mind, all late work is due on the 20th. I'm going to get your grades updated pretty soon so you can see what missing assignments you have. Anything that's going to be late, I'm just going to give you full credit if it's late, because you're all in different situations and you're all able to do different amounts of work right now. So for whatever reason, just get me all your stuff by the 20th. If it's an assessment, so this week I believe you have 2 assessments. You had one yesterday and then you have one coming up. So those are going to be graded for accuracy, but you're going to have the opportunity to redo and retake those probably as many times as we're able to. So I don't want you to be concerned about doing your best like the first time around. So like do what you can the first time around. I'll get those things back to you, learn from your mistakes, and then you'll have opportunities to resubmit those things. Okay, flip the page. So we're gonna go into how to solve genetics problems. So how to solve genetics problems. Okay, so they should, in the problem, they will tell you if it's simple dominance, codominance, incomplete dominance, multiple alleles, stuff like that. Because in the case of like codominance and incomplete dominance, they would have to tell you that, or else you don't know how to reference the phenotypes. So make sure that you're looking at that, just so you can determine like, is the heterozygote pink or is the heterozygote red and white, or like do you see both? Okay, so essentially I do a few steps in order every time I do genetics problems, just to make sure that I don't miss anything, but you will probably get to the point where you can do these in your head and just write the answers down, and that's totally fine. Whatever way works for you is fine. But there are 2 laws that we need to follow, and these are for multiple trait crosses. So we have genetics problems that are called single factor crosses, and single factor crosses involve 1 trait. So a single factor cross will just be 1 trait is being looked at. So we're just looking at plant color, or we're just looking at pea pod shape. And so when you do a single factor cross, all you have to do is you have to do a 4 square Punnett square. So if you see a Punnett square like this, that would be a single factor cross. If you're doing a multiple factor cross, So literally anything more than 1. That means that we're looking at a combination of traits. So we're looking at more than 1 trait. So maybe you want to see, like, what are the chances I have a child who has a widow's peak and who has hair on their knuckles and who has attached earlobes. So if I want to see the chances of like having multiple traits occur together, then you have to expand your Punnett square. So if you're looking at two traits at once, it actually turns into 16 squares, and then things get exponentially larger from there. You should never, ever, ever draw anything larger than a 4-square Punnett square, ever, okay? So I think in CP Bio, we like had you draw the 16 squares. We don't do that anymore, so there's 2 laws that we're gonna follow. But let's kind of review our Punnett square. So up here, what you put is you put the alleles for the sperm or the egg. So these are representing your gametes. So gametes go on the outside of your Punnett square. So it doesn't matter where you put the maternal or the paternal. It just matters that they're together. So let's just put our egg up here, and then we'll put our sperms down here. Okay, so let's say that for this factor, the father was heterozygous. So he's carrying a dominant allele and he's carrying a recessive allele. So like in his body cells, he is heterozygous. He has both of these. In his sperm, though, remember during meiosis, they separate. So meiosis ends up with haploid cells. So they only have one of each allele. So, sorry, this is the mother. So these would be representing her eggs. The eggs that this mother could produce will either have the dominant allele or the recessive allele. Let's say that the father was the same. So because he is also heterozygote, the sperm he produces, let's draw some sperm, could either carry the dominant allele or his sperm could carry the recessive allele. So these are just all the possibilities. When you fill them into the squares, so this egg would just be put into this box here. What this is showing, and then this sperm would go over here, this is showing fertilization. So we're saying if this sperm fertilizes this egg, what would the offspring look like? So these are all our possible offspring genotypes. We're not saying they're gonna have 4 offspring, but we're gonna tell them the chances that if they have an offspring, their offspring would have this genotype and phenotype. So then we could have another fertilization event down here where this egg is fertilized by this sperm, and you would get that offspring. And so when you're filling out these boxes, you're actually showing fertilization of the egg. So if you were a doctor and somebody asked you, like, what are the chances that my baby is going to be albino? Then you would look down here and you're like, well, I know albino is a recessive trait. And if both of you are not albino, and it's possible you're all carriers, meaning you carry the allele for albinism, then you can have a 25% chance of having an albino child. But if one of you is homozygous dominant, like big A, then you would have 0% chance of having an albino child. So oftentimes parents will ask, like, what are the chances? And so you can kind of see their chances within the Punnett square. So we're showing fertilization and possible outcomes. Okay, so if you get anything that is more than 1 trait, there are 2 laws you're going to follow. So the first law is called the law of multiplication. So the law of multiplication will apply if the 2 traits are independent of each other and the way that you're going to tell if they're independent of each other is if you see the word and. So if the question is asking, what are the chances that my offspring is tall and green, we're talking about plants, when you see that and, so you would take the probability of them being tall and you would multiply that by their probability of being green. Okay, so instead of having to draw a Punnett square with 16 squares to figure that out and counting all the squares, you would just do 2 4-square Punnett squares and figure out what are the chances of being tall, What are the chances of being green? The chances of being both of those things at the same time, you multiply those together, and that would give you your answer. We're going to do a problem with that in a second, so you'll see what I mean. The second law, which doesn't actually come up much in genetics problems, is the law of addition. So you only use the law of addition if the two traits you're looking at are mutually exclusive. Okay, and the word you're going to see here is the word or. So if you were asked what are the chances of rolling a 5, like on a dice, or a 6? These are mutually exclusive because if I roll a dice, if I roll a 5, it excludes my ability to roll a 6 because I rolled a 5. So having one of these excludes the possibility of having the other. Whereas with these, they're independent of each other because the probability of being tall does not exclude my ability to be green. Okay, so these two things can occur together at the same time. These two things cannot occur together at the same time. So if I wanted to figure out my probability of rolling a 5 or a 6, I would add them. So my probability of rolling a 5, there's 6 sides to a dice, so it would be 1/6. My probability of rolling a 6, there's 6 sides, would be 1/6. My probability of rolling a 5 or a 6, I would add those together. So it'd be 2/6, it'd be 1/3. So I have a third chance of rolling a 5 or a 6. Okay, or if it's like flipping a coin, what's the probability of flipping a heads or a tails? It'd be 1 because there's only a heads and a tails. Okay, we're going to do some genetics problems now. So I'm going to do problem set 1 first. And we're going to do number 1. Okay, so problem set 1, we're going to do number 1. I don't have the questions in front of me, so I'm going to kind of wing it. But in problem set 1, it's talking about the trait is skin pigment. So I usually just write out all my information. So my trait is the skin pigment. I have 2 alleles for this trait. So my 2 options for this trait are they could be albino or they could have pigment. I don't like it when they say normal because it makes it seem like albinism is abnormal. So these individuals could either have albinism or they could have pigment in their skin. So these are my 2 alleles for my trait. It says albinism is recessive, so I'm just gonna, you can choose literally any letter. And then it says having pigment is dominant. So those are my 2 alleles. It says 2 individuals that are both heterozygous, so heterozygous, are crossed together. And then it asks what are, I think it says something like, what are the chances of having a pigmented versus albino offspring? So let's just say like this is the mother, this is the father. When these cells undergo meiosis, remember these will separate. So when this individual produces eggs, she will produce eggs that have the dominant allele, and she will produce eggs that have the recessive allele. And then for this male, he will produce sperm that have the dominant allele, and he will produce sperm that have the recessive allele. So this is just showing meiosis, okay? We started with 2n equals, nope, let's not do that. We started with diploid cells, so they had 2 copies of every chromosome, and then we ended with haploid cells. They have 1 copy of every chromosome, okay? And so when you do your Punnett square, remember you just need to keep them together, so eggs go on one side, sperm go on another side, and then do your fertilization. Okay, so these are showing all the possible offspring. So if pigmented skin is dominant, then anything that has a dominant allele, because it's expressed over the recessive, will have pigment. So individuals that will be pigmented, that is 3. And then we say individuals that are albino, it's 1. So we say it's a 3-to-1 ratio, okay? Two other ways you can write this. You can say there's a 75% chance of being pigmented, 25% chance of being albino. So 1 divided by 4, 3 divided by 4. You could also do it as a fraction. So you could just do 3/4 to 1/4. If it's multiple choice, well, you're not going to have that on your AP test. But if you have that on one of my tests, it'll be written in one of those ways. If it's an FRQ, they'll probably ask you, or you could write it any of these ways and it would be correct. Oh, guest speakers here. Okay. Then we're going to kind of skip down and do number 5 because it's a 2-factor cross. She's going crazy. Okay. So in number 5, it kind of walks you through how to do the law of multiplication. I believe it has a 16 square Punnett square there for you, so I'll show you how to go through that. So fill it out this time. Going forward, you should never, ever, ever draw a 16 square Punnett square. Okay, so for number 5, there's actually 2 traits we're working with. So trait number 1 is the color of our seeds. So we have seed color. Trait number 2 is plant height. So now we're going to be looking at 2 factors, 2 traits at the same time. So for seed color, it says being green is dominant. So I'm just going to choose G. Being yellow is recessive. The key to picking your letters is just pick letters that look different from the capital and the lowercase. Like when you choose like a C or an S, sometimes it's hard to tell the difference between the two. So I just try to make sure I pick something that is just really different. For plant height, they said they could be tall, and tall is dominant, and then they didn't tell you the recessive, but you could just say dwarf or short. Just do the opposite of your dominant allele, and it said the other one is recessive. So I make my T look really different. Okay, then it says you're going to cross individuals that are heterozygous for both. So we're going to have heterozygous for seed color, and they're heterozygous for plant height. So this is one individual, and they're going to be crossed with another individual who's also heterozygous for both traits. To figure out the gametes that this individual would produce, you would have to find every combination of our 2. So you could just do factoring, F-A-C-E. So, is it F-A-C-E? I can't think of what okay. I don't think it is F-A-C-E. Never mind. So you would just have to find all your combos, right? So law of segregation states that this allele needs to go into one gamete. This allele goes into a different gamete. But then same thing here is like this allele needs to go into one gamete. This allele needs to go into a different gamete. So we just have to figure out all our different combos. So we could have a dominant allele with this dominant allele. We have this dominant with this recessive. That takes care of our dominant. We'd have this recessive with this dominant, and then we can have the recessive with the recessive. So this individual can produce 4 genetically different eggs. Same genotype here, so we're going to get the same gametes. But these would be like sperm. So this individual could produce 4 genetically different sperm. You would put all 4 of these on your Punnett square. So you would have egg, 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 sperm, 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 and you would draw a 16 square Punnett square. And you would fill in the squares with what is their genotype for seed color and then what's their genotype for plant height. That's crazy. So what we do is we do the law of multiplication instead. I'm going to draw this over here. So the law of multiplication, because we want to figure out what's the probability that their offspring are tall and green. So remember, just because the plant seed color is tall, or the plant is tall, does not mean its seeds can't be green. So we're not mutually exclusive here. They can be both at the same time. So if I want to know my probability of being tall, and these are my parents, I'm going to just do a Punnett square for my tall, and then I'm going to do a separate Punnett square for my green, and then we're going to multiply. So let's do green first. So my probability of being green, here's my eggs, here's my sperm, here's my fertilization, right? So my probability of being green is 3/4. Now let's do my probability of being tall. So here's my eggs, here's my sperm. So my probability of being tall, because remember green is dominant, tall is dominant. Probability of being tall is also 3/4. So my probability of being green and tall, you multiply those together. So it'd be 9/16. Yay! And then you don't have to fill in your 9 square Punnett square. Okay, so that's how to do those. So maybe like pause this now and then finish up problem set number 1. Problem set number 2 starts with codominance and incomplete dominance. So I'm not going to go over that. The only difference from this one would be how you do these ratios. So instead of just having 2 different phenotypes, you would have 3, because you would have the 2 poles and then what would be your heterozygote. So it would be 3 different things here, and what are the probabilities of being each of those. Same thing, it'd be the same for codominance and incomplete dominance. For problem set 2, I just wanted to go over how to do the blood typing. So we're gonna do number 4. So in number 4, it says the father of a child is type AB blood. So we're gonna say our male has type AB blood. So that's his phenotype. And then it says the mom, mistakes, it has type A blood. So these are their phenotypes. It says what blood type would their children not be able to have? So we have to look at their genotypes to figure this out. So to have type AB blood, remember blood type has 3 alleles, IA, IB, and the recessive I. Okay, so to have type AB blood, this is codominance. They have to be IA, IB. So that's the only possible genotype for that individual. For the mother, because she's type A, she has 2 possibilities. She could be homozygous dominant, so IA, or she could be heterozygous, IA, recessive I. Okay, so when we're looking at their children, we would just have to see like what gametes can this individual produce. So on our Punnett square, we know the father has to be IA, IB. Okay, we know the mother has at least 1 IA. So we're just going to kind of like fill in what we know. So they could have fertilization, a type A baby, fertilization. They could have a type AB baby. Then we just fill in what we have. So we have that and we have that. So now we're trying to figure out, so we know they could have a type A, they could have a type AB. Now we're trying to figure out, could they have a type B blood baby? Well, if this mother is heterozygous, so this would be a lowercase I, it's possible they could have a type B, right? Because right here, this would be little I. So they could have a type B baby. So we have 3 of our 4 blood types. Our last blood type is O. The only way to get O blood type is homozygous recessive, little I. So is it possible to get that? No, because we know the father is AB. So these are the 3 they can have. They cannot have type O. So a lot of the blood typing problems have you go backwards, okay? So things maybe to jot down for blood typing. There are 4 blood types. So these would be my phenotypes. And then the genotypes, there's going to be 6 of them, right? So for type A, they could be homozygous dominant or heterozygous. Oh, she meowing. For type B, same thing. They could be homozygous dominant or heterozygous. AB, this is our codominance, so I can have both. And then O is homozygous recessive. So as long as you know like all these possibilities, you can pretty much solve all of the blood typing problems. Okay, so you're gonna be working on those problem sets. I'll post the keys on Saturdays. We have office hours today, so if you have questions, come in for office hours. And I'll talk to you guys later. Bye.